All right. My name is Joe. I am a medicinal chemistry graduate student, and I am a graduate of Wake Forest University from Winston-Salem, North Carolina, which is where I'm from. And I'm really excited to be here studying epigenetics in the Tretyakova lab, because that's where my interests really do lie. As an undergraduate, I studied the enzymology of DNMT3A, and that really inspired me to pursue epigenetics as a grad student from the medicinal chemistry standpoint. And so I'm an interesting case where I'm co-advised between the Tretyakova and Pomerantz labs. And so my project lies in looking at the function of BPTF, which is an epigenetic reader of histone tail modifications. This protein is known to be heavily involved in the progression of pediatric neuroblastoma. And so one aspect of my project is leading an SAR campaign of inhibitors of this protein. And what's interesting in our collaboration with the Tretyakova lab is we are able to use native mass spectrometry techniques to really probe these ligand-target binding interactions. And this is an experience that really speaks to the collaboration of professors that study epigenetics in the Department of Medicinal Chemistry. So I'm very excited to be exploring many different facets of medicinal chemistry, from mass spectrometry techniques to NMR applications in the Pomerantz lab to SAR campaigns of these chemical probes that we can potentially use to treat disease. This is really important because epigenetics is a continuously evolving field when we look at it from a medicinal chemistry standpoint. There's so much that's unknown, and there's a ton of research that needs to be done and targets that need to be elucidated, so that way we can study these systems and get a better understanding of drivers of disease. The research environment in our lab is a very collaborative atmosphere between many people that have different expertises, and they can provide new insights and new perspectives on certain techniques that you're either trying to get off the ground or techniques that you're trying to optimize. So it's a very collaborative atmosphere in the sense that you're not afraid to ask questions. That's a key to science, is you're constantly asking questions and you're constantly improving protocols as you know it to get results, to get new findings. And I think that's something that can be said among many labs here at the U, is that it's a very collaborative environment among many individuals that have varying expertises, and they can offer new insights to your project. Natalia is very hands-on. I think she really prioritizes the students' learning experience. She really wants her students to not only get comfortable with the science, but learn new scientific techniques that will progress your research, as well as give you new skills that will enable you to succeed later in your career, whether that be as an academic scientist or an industrial scientist. I think she does a fantastic job of ensuring that you're learning these techniques along the way and not just performing an experiment to get a result. And I think my collaboration with the Pomerantz lab really speaks to that, where I'm learning tons of new techniques, NMR-based techniques, chemical biology-based techniques that are going to be benefiting projects in both labs. As a first-year graduate student, I am not entirely sure, but ongoing study in the field of epigenetics. So I'd really like to continue looking into elucidating new epigenetic targets that are currently unknown. This training that I am receiving in the Tretyakova lab will set me up very well for a career in industry, just because of the breadth of knowledge that I will obtain during my time here. So I'm really looking forward to entering the medicinal chemistry industry, particularly in the field of epigenetic inhibitors. University of Minnesota is special because it is such a collaborative environment. There's many professors that are doing similar fields of work, but they do have slightly different expertises, and these can combine to really drive projects and new findings. Other people should consider joining the Tretyakova lab because there's so much in terms of techniques that you can learn. Obviously, one of them being mass spectrometry techniques. There's also many techniques that you can think of in terms of the synthesis of new compounds or chemical biology assays that really drive SAR campaigns. There's a lot of cell growth assays that you can do, so cell culture, if that's something you're interested in, and even looking at how small molecules impact the expression of genes or proteins, so doing genomics and proteomics experiments. There's really, the sky's the limit in terms of what you can do in the Tretyakova lab, and so I think it's a good fit for anyone.